Welcome to the second PowerPoint of Unit 8. May the odds be ever in your favor. Quotation from Ralph Waldo Emerson, the transcendentalist. Every man is a quotation from all his ancestors. And here we have an actual image from Queen Victoria's family tree in 1901. Kind of an interesting piece there. So our learning goals here, we're going to predict potential offspring. That predict verb is really important because that's the difference between a book report and science. We're making predictions. And we're going to use Punnett squares. Now, we already know that we each have a homologous pair of chromosomes with two different alleles on them, and that our parents give one of their alleles at random through a gamete, whether it's a sperm from the father or an egg from the mother. Now, in our lab, we learned that random chance results in predictable ratios. That statistically, you'll get a good ratio with larger sampling. And the odds predict what will occur. Now, they predict by odds. We can't say with certainty that this mother and this father, but we'll get there. So we're talking about sexual reproduction. So we were talking about Mendel earlier. He was still working with the process of sexual reproduction, but in plants. The plants are convenient because he could control which two organisms were crossing with another one. So what he did was he removed the male parts of the plants, which are called the stamens, and then that way he could use a small tool to move the pollen from one plant to another. That way it didn't accidentally get fertilized by a different plant or this or that. He had a lot of control there. And so he could generalize those results to all organisms that undergo sexual reproduction. So whenever that happens, we call that a cross. So he selected 7 traits and he picked dominant traits. Now he only took a finite amount of data. Now, you can see some of them are in the thousands and some are in the hundreds, but with those data points, if you look down all the ratios, 2.96 to 1, 3.01 to 1, 3.15 to 1, 2.95 to 1, they are not exact and perfect because we did not have a perfectly large sample of data. We had a small sample of data, which is imperfect, so we get a ratio that's imperfect. We can see that we're already getting really close. If we had an infinite number of data points, which is impossible, we would expect a perfect 3-to-1 ratio. So that gave rise to Mendel's first law, the law of segregation, which tells us that organisms get 2 copies of each gene, 1 from each parent. Now, only 1. Now, what that led him to understand, that all came from this statistics and this ratio, is that gametes only get half of the copies that a parent has. A parent has 2 different ones they could give, but they only give 1 of them. The next question that's obvious from there is the law of independent assortment. Which gamete goes with which gene? It's random, meaning that there's not a dependence on another variable that controls it. It's independent. So we keep using this word gene. Specifically, the gene is a chunk of DNA. The DNA that got wound up around the histones to become a chromosome. In that DNA, a specific length of it, sometimes shorter, sometimes longer. Different genes are different lengths. But however many sequences it is in the DNA, that set of sequences codes for 1 trait. And that's what we call a gene. But not all genes are the same. Even a specific gene that encodes for a specific trait, the gene that I have is likely different than the gene that you have. So we identify them based on their locus. As you know, it sounds very similar to the word location, because that's what locus means in Latin, is place. So it's found in a very specific spot in the DNA. So if you look at a specific spot of the chromosome, there's a spot where it got wound up, that if we unwound it there, we unwrapped it around those histones, and we looked at this sequence, that's where the gene's found. Now, different versions of different genes we refer to as an allele. We used that word earlier. It's actually a shortened form of the word allelomorph, which I think is a pretty cool word, but I guess it is a little bit of a mouthful. It comes from two Greek words, allele, which means other, and morph, form. So allelomorphs are just other forms of the same thing, so which we shorten to alleles. So they're other forms of genes. So the gene that's found at the specific locus on the chromosome encodes for one trait. Now, we don't all have the same DNA at that locus. Our genes are different from each other. That's the allele that we're talking about. We saw before that some traits are more readily passed on than others. We refer to those as dominant. And some are, more likely to be overridden. Those are recessive. So we end up with these two different types, the genotype and the phenotype. The genotype refers to the alleles we have, the other forms of the gene. The phenotype refers to how those genes are expressed into traits. Pheno, in this case, actually comes from the Greek phenos, which means to appear, to show. So the genotype are the alleles we have, but the phenotype is how those alleles are manifested in what we can actually see. Now, we've said this before, science is about predicting, not just describing, not just making reports, not merely observation, but to use that information to form a model that we can predict other things with. So, what are we predicting here? How is this science and not just descriptions? Well, if we have a cross between two parents, what is the probability that a certain trait will be passed on and show up? That's something we can predict. Bill Nye's on board. So let's talk about a sample cross. Let's say that for a certain trait, any kind of trait, this is a trait that the father has a dominant and a recessive allele. It means that the father has this trait. He's carrying the recessive allele, but that dominant allele is there. So we represent dominant alleles with a capital letter and recessive alleles with a lowercase letter. Just because it's simple, it's easy, and we like things that are simple and easy. So his genotype is capital A, lowercase a. So when our father's sex cells go through gametogenesis and we get that sperm, we know that in meiosis, whenever the sister chromatids are split, that the law of independent assortment says that it's random which gene goes to which side. But there's only two options. Either that sperm got the dominant allele capital A, or two, the recessive allele lowercase a. Now in this situation, let's say, for this example, the mother also has a genotype of capital A, lowercase a. That means that she is also expressing this trait. So her egg could have either received the dominant allele or the recessive allele. So what if we took the sperm from the father and the egg from the mother and combined those to form a zygote? What's the most likely phenotype? Well, most likely we're going to end up with a offspring that shows the same traits that both parents had. Both parents exhibited that dominant trait. And if we look at the three options, there's 2 out of the 3 are exhibiting that trait. They have at least one dominant allele. But let's look closer. So if the egg has the dominant allele and the sperm has the dominant allele, we end up with a genotype of capital A, capital A. Now, if we have the same egg, the dominant egg, but the other recessive allele in the sperm, then the second option is capital A, lowercase a. But now, if we take the second egg that has the recessive allele and we combine it with the first sperm that has the dominant allele, we get capital A, lowercase a again. It's actually the same thing as the second pairing. The last option is, what if they both give recessive alleles? So there's 4 different options. If we look at the genotypes, 1 out of 4 options ends up in capital A, capital A. 2 out of 4 options is capital A, lowercase a. And 1 out of 4 options is lowercase a, lowercase a. There's a 50% chance, the best chance, that you end up heterozygous, just like the parents in this case. There's only a 25% chance you end up with dominant-dominant, and only a 25% chance you end up in recessive-recessive. That's a 1 to 2 to 1 ratio. But if we're looking at phenotype, we end up with a 3 to 1 ratio, because in order to express this trait, you only have to have at least one dominant allele. 3 out of 4 options had at least one dominant allele. So 75% of the children, or we should say each offspring, has a 75% chance of ending up with that phenotype, and only a 25% chance of ending up with the recessive phenotype being expressed. Now that was a lot of work and organization. There's an easy way, a shortcut. It's a graphic organizer. It's called a Punnett square, named after Reginald Punnett, who might have written the first popular genetics book that was ever released to the public in the early 1900s. So what this allows us to do is, if you see on the top, we have the father's genotype, capital A, lowercase a. On the left, the mother's, capital A, lowercase a. And if we look at the 4 boxes, there are our 4 options. And you can see how easily it relates to the outside. So let's dive into a developing level question. Describe the phenotypes of the parents and the children as either dominant or recessive. The father, in this case, has at least one dominant allele, which means his phenotype is that of the dominant trait. The mother does not have any dominant alleles, so her phenotype is that of the recessive trait. If we look at the options in the boxes of possible offspring, we see that 2 out of 4 possible offspring exhibit the dominant trait, whereas 2 out of 4 offspring have a phenotype of the recessive trait. Now let me make this point absolutely clear right now. The Punnett square does not mean that the mother and the father had 4 children. Inside the boxes are possible offspring. They show us odds. So the odds are, for each offspring they have, there are 2 out of 4 options that have a dominant phenotype, a 50% chance, and 2 out of 4 options that have the recessive phenotype, a 50% chance. Okay, what are the possible genotypes for the children of this cross, and what are the odds of each of them occurring? Well, we can see there's only 2 possible different genotypes, capital D, lowercase d, or 2 lowercase d's. So that means there's a 50% chance of each. Okay, mastery question. Compare and contrast the potential results of the following crosses. We have 2 different Punnett squares. They have both the father on the left and the father on the right have the same genotype, but the mother on the left is heterozygous and the mother on the right is homozygous. So on the left, we have 2 different kinds of genotypes that show up, either homozygous dominant, capital A, or heterozygous dominant, capital A, lowercase a. But that means on the left we only have one kind of phenotype. All of them are still exhibiting the dominant trait. On the right, we see that there is a 100% chance that this offspring is going to be homozygous dominant, capital A. So on the left and on the right, we have 100% chance in both cases of getting the dominant trait in our phenotype, but we have differences in genotypes. So a test cross is an idea of, what if we knew one of the genotypes, but we didn't know the other. What if we did a cross and then looked at the ratio of offspring and then used it to kind of work backwards and figure out the genotype? Here's a good example. Let's say we know that the father's genes, that the alleles are heterozygous dominant, capital A, lowercase a. And then let's say we know that from this test cross that whether these are two animals or two plants or whatever, we did a bunch of crossing, we got a ratio that looked like this. That we got a 3-to-1 ratio. All dominant, some homozygous, some heterozygous. What does that let us conclude about the mother? Well, if we look in the top left box, the father donated a capital A. Well, if the offspring got a capital A, then the mother must have donated the other dominant. And if we look in the lower left, looks like the mother donated either a dominant or a recessive. So it looks like we can conclude that we probably didn't have a large enough sample size to get good ratios. Okay, what if we did more samples? We had enough data collected that we started to notice, wait, there are some that are homozygous recessive. Here's that 1 to 2 to 1. So we can conclude that the mother's genes are probably capital A, lowercase a. So our Punnett squares so far have had 4 outcomes because you had 2 alleles from each parent. 2 times 2 is 4. How many outcomes would a Punnett square have that was considering 2 pairs of alleles? 2 genes at the same time. So let's say one gene is going to be represented with R and one with Y. So look at the top left cross in the Punnett square. We can see we had a capital R, lowercase r, a heterozygous dominant, and a mother with the same. We get that 1 to 2 to 1 ratio. 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. So if we look at the 3 options there in the middle, we're 1/4, half, and 1/4, and then what if those cross with the results of the Y's? So there are the Y's, we have that same 1 to 2 to 1 ratio. So if the homozygous RR was selected for R and the Y is selected for Y, then we end up with dominant R homozygous, dominant Y homozygous. That the offspring will get both of those traits homozygous. If we keep doing that, for each option that comes out of the R Punnett square and each option that comes out of the Y Punnett square, we see 9 different options. But remember, the middle section is 50%. So we get twice as many of them as the others. So that's why the lowest common denominator of fractions is 16, because it's 2 times 2 for the R's and 2 times 2 for the Y's. So 2 times 2 times 2 times 2 is 16. So that's why we're getting these 16ths coming up. So draw 2 different Punnett squares, even if they end up being very similar, that have a 3 to 1 ratio of phenotypes. So you'll need something heterozygous. Okay, I'll give you a second. You can pause. Why don't you fill out this dihybrid cross? There it is, all filled out. We're just copying the top and the bottom together, just like the same thing we did with the 2x2, but just more. So using that same Punnett square, what's the percentage chance of seeing each genotype? And then what's the percentage chance of seeing each phenotype? So we have a lot of different genotypes going on. We're all out of 16. Some of these can be reduced, like 4/16 is actually 1/4. Whenever we look at the phenotypes, though, 9 of them are dominant in both. 3 are dominant in A and not in B. 3 are dominant in B and not in A. And 1 is not dominant in either. This is a common pattern that we see emerge. Okay, let's draw a dihybrid cross that has a 1:1 to 1:1 ratio of genotypes. Think about that for a second. Alright, so I used S's and Y's. You can use whatever you like. Now if you count up, I think you'll see that it's 4 to 4 to 4 to 4. I counted 4 of each, but that reduces to a 1 to 1 to 1 to 1. Proficient question. Why is it important that sexual reproduction result in offspring that do not have identical traits to the parents? Well, it's important because in evolution we need variation in our population. We need some members to have some advantages while other members have different advantages. That way it depends on what the situation is. Sometimes person A has an advantage in that situation, but in a different situation, person B has an advantage. We need to have variation.